For decades, we've been totally fascinated by this really simple, powerful idea that somewhere hidden in our DNA, there's a little set of longevity genes that basically control how long we live. The promise was, hey, if we could just find them, flip the right switches, we could unlock the secrets to a longer life. Well, today, we're going to look at how one incredible experiment with fruit flies completely turned that whole idea on its head.
Okay, so let's just dive right in with the big question, the one that's been at the heart of aging research for years. Is getting older really governed by just a few super powerful genes? Or is the story, well, a lot more complicated? This was the mystery that scientists were determined to solve.
To really get why this was such a breakthrough, first, we've got to understand what most scientists believed before, say, 2010. The whole community was pretty much focused on this massive hunt for these so-called master genes.
This old way of thinking was called the neoclassical model. It sounds fancy, but the core idea was that evolution and aging happened because of these rare but really potent genetic changes. Like one single mutation having a huge, noticeable impact. And this model made researchers think, okay, if we can just find these few key genes, we've cracked the code of aging.
So here's maybe the best way to picture the two competing ideas. The old view, let's call it the broken parts theory, saw aging like a car with a couple of bad components. You just find the busted spark plug, you replace it, and boom, the engine runs better. But the new idea, the orchestra theory, paints a totally different picture. It says aging is like a symphony. A long life isn't about one-star soloist hitting a perfect note. It's about the subtle, coordinated adjustments of hundreds of different instruments all playing together in harmony.
All right, so you've got these two big ideas. How in the world do you test them? Well, you run an experiment in evolution. And to do that, scientists turn to a creature with a super fast life cycle, the humble fruit fly.
The setup for this was just brilliant, so simple. The researchers made two different groups of flies. The A populations, they were forced to have babies really early in their lives. The C populations, though, were only allowed to reproduce late in life. This effectively selected for flies that could just live longer. And they let this go on for hundreds and hundreds of generations, creating two groups that were basically programmed for totally different lifespans.
And the result? Oh, it was dramatic. It worked exactly like they thought it would. After all those generations, the A flies, the ones bred to reproduce early, were living only about 60% as long as the C flies, the ones bred for a long life. The experiment was a huge success. They had officially evolved two different ways of aging.
And this right here is the million-dollar question. What was different in their DNA, right? If that old broken parts theory was correct, you'd expect to find just a few big glaring genetic differences, those specific longevity genes that the long-lived flies must have. But that is not what they found, not at all.
What the DNA sequencing showed was a total shocker, and honestly, it sent ripples through the entire field of aging research.
So what you're looking at here is called a Manhattan plot. Every single dot represents a genetic difference between the two groups of flies. The higher the dot, the bigger the difference. Now, if the old longevity gene idea was right, we'd expect to see just a couple of massive skyscrapers shooting way up past that red line. Just a few powerful genes doing all the work.
But just look at it. There are no isolated skyscrapers. What they found instead was more like a dense city skyline with hundreds of smaller buildings spread out across the entire genome. Hundreds of tiny, but statistically significant genetic changes were all pitching in to create that longer lifespan. This was the big aha moment.
And this just proved it, brilliantly, that the orchestra theory, or what scientists officially call the balance framework, was the right way to think about it. Longevity isn't about a few star players. It's this incredibly complex trait, a delicate balance of hundreds of genes with small effects all working together. The old model, the neoclassical one, of a few big genes, it just couldn't explain this data. So, what does this all mean for that original quest? It means this single experiment basically tore down the whole myth of the all-powerful, single longevity gene. So why is that hunt for a single gene so misguided? Well, for one, genes that have a huge positive effect on lifespan, they often come with downsides, so natural selection actually tends to get rid of them in the wild. Second, their effects are super unreliable. A gene that helps a lab mouse live longer might do absolutely nothing in a different mouse. And here's the kicker. These so-called longevity genes actually showed up less often than you'd expect by pure chance in these evolution experiments.
The whole way of thinking shifted so much that the researchers basically wrapped it up with this powerful statement. What they're saying is, look, studying these rare, big-effect longevity mutants in the lab, it's not really helping us understand how aging actually works in real, genetically diverse populations, like us.
And what's so fascinating here is that this discovery, it's not just about aging. It's really a symptom of a much, much bigger revolution that's happening across all of biology right now.
This quote just nails the shift in thinking. The old idea that we could map out biology like a simple flowchart, one gene makes one protein that does one thing, that's basically dead. We've opened up the black box of the cell, and what we found inside is just way more intricate than we ever imagined.
So the new reality of biology in the 21st century is complexity. For almost every single trait you can think of, from your height to your risk for disease to how you age, we're not looking at a simple A to B pathway. We're looking at a huge, interconnected network of genes and proteins and all kinds of molecules interacting with each other and with the environment. It's a whole system.
And to really put this in perspective, this shift to genomics is being called a refounding of biology. We're talking about a change on the same level as Darwin discovering evolution in 1859, or Watson and Crick figuring out the structure of DNA in 1952. We are in a brand new era, moving from a time of trying to reduce everything to its simplest parts to a new age of embracing biological complexity.
So this brings us right back to where we started. If there's no magic bullet, no single longevity gene we can just switch on, what does that mean for us? Well, it means the dream of a simple anti-aging pill is probably just that, a dream. But it opens the door to a much more powerful and frankly realistic understanding of health. One that focuses on the entire orchestra of our biology, not just on one broken instrument. The challenge for us now is learning how to be a better conductor,